Now we are going to discuss about the process of spermatogenesis. Spermatogenesis means the formation of haploid sperms from diploid spermatogonia. The formation of haploid sperms from the diploid spermatogonia is called spermatogenesis. Generally, in human males, the spermatogenesis will start at the age 13 to 14 years. We'll call it as generally puberty. So at the puberty age, it will start and it will continue throughout the life. The spermatogenesis will take place in the seminiferous tubules. In the process of spermatogenesis, there are 4 different stages are present. In that, the first one is multiplication phase. Second one is growth phase. Third one is maturation phase. And the last one is differentiation phase. As we discussed before, in the seminiferous tubule, a single layer of cuboidal epithelial cells are present. These cuboidal epithelial cells are called germinal epithelium. From these germinal epithelial cells or the cuboid epithelial cells, the sperm's production will take place. In between these germinal epithelial cells in the seminiferous tubules, there are the tall, large-sized, another type of cells are called Sertoli cells. These Sertoli cells are useful for the nourishment of producing sperms. Among the 4 different stages of the spermatogenesis, the first one is multiplication phase. During the multiplication phase, these germinal epithelial cells will undergo into repeated mitotic divisions to produce more number of cells. These more number of cells are one attached to another one. So these more number of cells will develop due to the repeated mitotic divisions of the germinal epithelial cells. Hence, it is useful for the production of millions of sperms. After the repeated mitotic divisions, some of the cells will ceases the mitotic division and starts growing in size. These abnormally grown sizes are called primary spermatogonia. So, the stoppage of the mitosis division and the growing in size and reaching almost double in size in compare with the spermatogonial cells is called growth phase. So in growth phase, there is no division, only growing in size. So the primary spermatocyte size almost will become double in compare with the spermatogonia. After growth phase, this primary spermatocyte will undergo to the first meiosis division, and after first meiosis division, it develops 2 cells. These are called secondary spermatocytes. Immediately, these secondary spermatocytes will undergo into the second meiosis division. The second meiosis division is equal to the mitosis or the equational division, and it develops into the haploid spermatids. So the production of spermatids from primary spermatocyte with the meiosis 1 and meiosis 2 occurs in the maturation phase. After maturation phase, from the from the spermatid, from each spermatid, the sperm will develop. So, the sperms are also consists of the haploid number of chromosomes. The production of sperms from the spermatogonia is called spermatogenesis. Spermatogenesis, and then the production of sperms from the spermatids is called spermiogenesis. After this spermiogenesis, these sperms are immature and non-motile, and they will attach to the Sertoli cells. The Sertoli cells are useful to nourish the sperms, and after nourishment of the sperms will detach from the Sertoli cells. The detachment of the sperms from the Sertoli cells is called spermiation. So, the spermatogenesis means the production of sperms from the spermatogonia. Spermiogenesis means the production of sperms from the spermatids, and the spermiation means the detachment of the sperms from the Sertoli cells. Here, it is a schematic representation of the spermatogenesis. In the schematic representation of the spermatogenesis, here the first cells are called spermatogonia. These are in diploid state, and after multiplication phase, all these, how many number of spermatogonia are developed, all these are the in diploid state, and then after multiplication phase, in the growth phase, the primary spermatocyte is developed. It is also in diploid state of chromosomes, R2, a number of chromosomes are present in the primary spermatocyte. But after a primary spermatocyte, after meiosis division, what and all cells are developing in the spermatogenesis? They are secondary spermatocytes, spermatids, and sperms are have the haploid set of the chromosomes. Moreover, from each spermatogonia, many number of spermatogonia are only developing. Next, from each spermatogonia, only one primary spermatocyte is developing, because the primary spermatocyte is developing from the growth phase. Next, from each primary spermatocyte, the two secondary spermatocytes are developing. From each secondary spermatocytes, two spermatids are developing. Hence, from each primary spermatocytes, four spermatids are developing. From each spermatid, one sperm is developing. So that from each primary spermatocyte and spermatogonia, two secondary spermatocytes, four spermatids, and four sperms are developing. Next, from each secondary spermatocyte, two spermatids and two sperms are developing. But from each spermatid, only one sperm is developing. Hence, if at all we are considering how many number of primary spermatogonia are and the secondary spermatogonia and the spermatids are required for the production of 100 sperms, during such timings we can calculate in such a way that for the production of 100 sperms there are 25 primary spermatocytes, 50 secondary spermatocytes, and 100 spermatids are required. To complete entire this process of spermatogenesis, it takes approximately 64 days. After this spermatogenesis, the sperms are immotile and immature. These sperms will enter into the epididymis through the vasa efferentia, and then in the epididymis and as well in the vasa deferens, these sperms will store approximately 30 days. During that time, these sperms will acquire the motility and maturation.